Hi guys, welcome to my channel. Today I discuss the history of plant tissue culture. Let's start. Firstly, introduction of plant tissue culture. Tissue culture is the in vitro cultivation of plant cells or tissue under the aseptic condition in the liquid or semi-solid nutrient media for the production of primary or secondary metabolite and regenerated the plant large quantity. Plant tissue culture is based on two unique properties of plant, totipotency and plasticity. Toti means to tell, and potency is the ability of the plant cells to develop whole plant from a single plant cells. Plasticity means ability of the plant cells to adapt in different conditions. There are two metabolites, primary and secondary. And primary metabolites means a primary metabolite is a kind of metabolite that is directly involved nor is normal growth, development and reproduction. Some common example of primary metabolites include lactic acids and certain amino acids. Next is secondary metabolite. Each are generally defined as small organic molecules produced by an organic organism that are not essential for their growth, development, and reproduction. Some common examples of secondary metabolites are toxin, gibberellin, and alkaloid, etc. Next is advantages of plant tissue culture. Disease-free plants and biogenetic pathway tracing, regular and uniform supply of raw materials, overcome in the fluctuation in quality, crop improvement. These are advantages of plant tissue culture. Next is disadvantages of plant tissue culture. Cells may lose some differentiated characteristics, hard to maintain, only grow small amount of tissue at high cost and differentiation. Next is history of plant tissue culture. Gottlieb is called father of the plant tissue culture because in 1902 uh, he uh, he first attempted the plant tissue culture, and his method is called the totipotency method. Um, but he but he not the not the use this technique because this time not discovered the um, hormone. Gottlieb Haberland discovered the cultivation of mesophyll cells and epidermal cell in this flower. This is a Trident scantia species. This is Ornithogalum, and this is Acornia crespia species. And Kotler and Robin. In 1922, they successfully culture root and stem tip respectively. Next, Professor Henning. In 1904, Henning tried embryo culture in Cruciferous species. Next is Professor White. In 1934 to 39, is successfully cultured tomato root tip in artificial culture media. Thereafter, he gave it successfully and subculture in the fresh media. And the media content in organic salt, sugar, yeast extract. Next is Guthrie and Wheat and Novagood. 1939, they established culture tomato root tip in they develop callus and maintenance, whether callus is a mass of undifferentiated cell. Next is Professor Wendt. In 1926, he discovered IAA, means indole acetic acid, which is a plant growth hormone. This is auxin. This picture is oxygen picture. The next is Van Overbeek. 1941, he established the nutrient value of coconut milk by cutting the embryo. In the embryo culture, he developed the seedling, and during the development, he used endosperm along with inorganic salt, mineral salt, sugar, vitamins in the artificial nutrient media. Next is Ernest Arbe Bell. In 1946, stem tip culture and organ culture were established to produce whole plant. He developed the method for extraction in growth of the extract plant part. He isolated the important part of mesometic cell from which the whole plant can be developed. This method was thereafter being used in industrial scale for the plant propagation. Next is Mori. In 1953 to 54, he developed the plant from the single cell after the successful of the Kelas culture from the X6 plants. Next is Miller and Cook. In 1957, demonstrated the hormonal control plant, and they also found out that amount of oxygen is more in the nutrient media as compared to cytokinin. Then root formation takes place, and if the amount of cytokinin is more is the nutrient media, then oxygen then should formation take place. Next is Reinhardt and Stewart. Somatic embryogenesis was done. Somatic or non-reproductive cell used to form embryogenesis, and their carrot suspension culture was used. Next is cooking. 1960, protoplasts was isolated. Protoplasts are the cells without cell wall. So cooking extracted many cells. After that bioenzymatic degradation, protoplast was removed. Next is Bergman. In the same year, 1960, the Bergman developed the plotting technique cloning. Next is Murasaki and Cook. 1962, developed media for the Kelas culture where they increased the concentration of inorganic salt. MS media naming was done. Next is Guha and Maheshwari. 1964, first developed the haploid anthraculture. And first anthraculture plant is Dhatura. Power. In 1970, protoplast fusion was done with inter and intraspecific fusion. And Tekabe, 1971, developed the plant from the protoplast, which plant were developed. Next is history of plant tissue culture, Carlson. In 1972, first intersomatic hybrid was developed, protoplast fusion. In this experiment, two plants from different species were taken and somatic cell isolated and fusion together by protoplast fusion. Here, Nicotina gluca and Nicotini lens turbe were scattered, and their somatic cell were isolated, and isolated somatic cell were then treated with enzyme to remove cell wall of the protoplast were then fused. After the fusion of protoplast, the hybrid that is formed is called somatic hybrid. Next is Silton et al. In 1977, introduced the TI plasmid into the plant. This picture is a TI plasmid picture. And last is Hers. In 1984, developed the Tanzanic tobacco plant. And this is my reference. And thank you. If you like my video, then support me. Thank you.